you hear? Can you hear?
Yeah, I can hear you. Am I audible to you?
Hello? Can you hear, madam?
Hello?
Yes, madam.
Yeah, I can hear you. Are you getting
Thank you.
my voice over there?
Yes, madam. Audible.
Yeah. So, what I'm thinking is I'll probably switch off my video because I'm not sure. Yeah, because I'm not sure if the load on the internet connection here would be too much otherwise.
Please.
Yeah,
Sure,
I'll switch off my video. Yeah, I'll be
sure,
like, my mic is on, but I switched off my video.
sure, madam. Sure.
Yeah.
First of all, very thanks, madam. Thanks and gratitude for your wonderful support towards the development of our students' career community. Thank you, madam.
Uh, Thank you for having me here. I mean, it's my pleasure to be actually a part of this educational series.
Thank it is அனைவருக்கும் இனிய காலை வணக்கம். It is an honor and privilege for me to welcome you all on the behalf of Arulmigu Kalasalingam College of Pharmacy for this 55th International Virtual Learning Series with the blessing of Almighty, our founder Kalvi Vallal Devathiriti Kalasalingam Ayya, with the support of our chairman Ilaya Vallal, Dr. K. Sridharan sir. I am here to deliver the welcome address for this uh, 55th International Virtual Learning Series in association with the Indian Pharmaceutical Association, South Tamil Nadu local branch, head office at Arulumigu Kalasalingam College of Pharmacy. Today we are proud to have an eminent scientist, a well-known researcher, and honorable resource person, scientist Dr. Akriti Srivastava, a research scientist, Department of Neuroscience, USA, to grace this occasion. Let me acquaint you with the sterling quality of this eminent personality, who is a brilliant researcher and undisputed researcher. It is great pleasure for all of us that she could find time to be with us in spite of her busy schedule. I take this opportunity to convey my sincere thanks to you and your esteemed organization for accepting our request despite of your busy schedule for being a valuable part of this session, madam. Welcome, Dr. Akrit, madam. Most welcome, madam.
Thank you so much, Dr. Gnana.
Sir, next, I welcome all the professor, faculty members, and my dear enthusiastic students for today's session. My dear students, this session hope will enrich your knowledge and update you about the scope of stem cell research in pharmaceutical sciences. This is the first and foremost needy for your career. Kindly utilize and grab the great opportunity and explore your knowledge. All the very best, my dear students. Once again, I welcome you all. Thank you. Thank you. Welcome, Dr. Akrit, madam. Thank you.
Thank you so much.
Please continue your presentation.
Okay. Yeah, thanks a lot for having me here, and it's really my pleasure to be a part of this educational series and contribute in whatever way possible to the possible research career of the students here. So I'm gonna switch off my video to avoid any interruption in the internet
Sure,
services here. So I'll just begin with the slides straight away. Yeah,
sure.
yeah, sure. So, as you might all know by the introduction, like today's topic or today's talk is going to be on the scope of stem cell research in pharmaceutical sciences. And I'm sure all of us, like everybody present here, must have heard about, must have some vague idea about stem cells, if not in like detail. So let us see what stem cells are. So term stem cells was actually coined by Bob Ryan Hacker to define a group of cells that could give rise to an entire germline of an organisms, right? And these cells are said to be completely naive, completely uneducated cells, which can be stimulated to be converted into any type of cell in the body that we have. So based on the specific stimulus you give, you are you can differentiate these stem cells into, say, liver cells, kidney cells, nerve cells, or even heart cells, and so on and so forth. And the biggest advantage with these cells is you can easily derive them from either adult body tissue or even from the embryo. So another method of generating them is through reprogramming. So, what do I mean by reprogramming? Reprogramming is like when you take the cells from an adult tissue and then you add certain external factors, which we call as the stem cell factors, and convert these adult tissue cells back to their origin of like back to their original stem cell stage. That process is called reprogramming. Now, this I'll be covering later on in the slides, but this is one of the major advantage and major discovery that has been made in the stem cell field, which has made their use far more easier. And also, they have this inherent capability of self-renewing or self-replicating, which makes them a complete living system in themselves. Now, what are the types of stem cells? So broadly, the stem cells have been divided into various types based on the potency of their potency. So by potency, I mean their ability to differentiate into several other cell types of the body, or in fact, they have the ability to differentiate into almost all the cell types of the body. So based on that, we have totipotent cells, pluripotent potent cells, and multipotent stem cells. Now, totipotent cells, stem cells, they have the capacity to give rise to all cell types as well as the extra embryonic tissue like placenta. So in a way, they have the ability to completely form a whole organism. So that's why zygote, the fertilized zygote that we have, that qualifies as a totipotent stem cell because that leads to development of a complete organism. Second are the pluripotent stem cells. Now, pluripotent stem cells can give rise to all the possible cell types of the body, and they are basically of two types: the embryonic stem cells as well as the induced pluripotent stem cells. Then comes the multipotent stem cells, which have a restricted ability to differentiate into just a limited number of cells. So they're basically the adult stem cells, which can be mesenchymal stem cells, neural stem cells, or the hematopoietic stem cells. So I'll be taking them like briefly, but since my focus on today's talk would be on the clinical applications or the uh, applications of stem cells in pharmaceutical sciences. So I'd basically be focusing on embryonic stem cells and induced pluripotent stem cells. Now, embryonic stem cells, they are derived from basically from the embryos, which are usually the embryos that are used here are usually the discarded embryos from the IVF process or the in vitro fertilization process, which are no longer used by the patients or the clients. So these cells are essentially derived from the embryo. They, like any other stem cell, have the ability to self-renew or self-replicate and have the capacity of giving rise to all the possible cell types of the body when they are stimulated with a specific stimulus. And the first human-based ESCs or embryonic stem cells were reportedly found in 1998. And since then, they have been used for various studies based on deciphering the disease mechanisms in humans or for drug screening. But what limited their use or what was the ethical concerns associated with these cells? Now, by ethical concerns, I mean various bodies across the globe protested about their use in research because it is derived from an embryo, which is like a human embryo, which is a living human, essentially, and you can say a part of a human life, right? So the argument was that since it's derived from a human embryo, which is, even though it's not been implanted in a woman's body, but still it is a living thing and of human origin. So there were a lot of protests across the globe about their use. So they're marked by a lot of ethical concerns. That this, because of this, there was the use decline. I mean, people were slightly skeptical in using them for various research purposes because of the backlash they were receiving. Additionally, these cells also have this inherent ability of tumor formation. So later on, you never know when that tumor could turn malignant or when that could be detrimental for these cells as such. So these two factors played a major role in the limited use of embryonic stem cells in the pharmaceutical industry or in the disease study or the drug screening process in general. So because of all this, there was a lot of confusion among the researchers, like as to how to go about the use of stem cells, since they had so much potential in them for, for in like any field of biomedical research. So then it was only when the induced pluripotent stem cells came into picture that the stem cell research, the whole face of stem cell research, or stem cell research or stem cell biology kind of changed for everybody. So this was a discovery made by Shinya Yamanaka in 2006 in Japan. After a series of failed experiments and a lot of patients, he was able to convert adult cells or adult tissues from a person back into the stem cells by introducing certain stem cell factors, which are also named after him, called as the Yamanaka factors. So by introducing those factors into a normal adult tissue, he was able to convert these cells back into the stem cells. So, and because of that, he was awarded a Nobel Prize in 2012 because it was such a huge discovery and a boon for the whole biomedical research fraternity. And when I say induced pluripotent stem cells, I literally mean that these are the types of cells which can be externally stimulated to convert or to give rise to every possible kind of cell type in our body. And these cells, these stem cells, found were uh, came out to be a really important tool in disease modeling, and especially when it came to identifying the mechanisms behind certain human diseases, and also in the process of screening drugs or potential compounds which could be used as drugs in the pharmaceutical industry as a whole, because these were essentially human derived and they didn't have the problems that were associated with the embryonic stem cells. They didn't have those problems. There were no ethical concerns associated with them because they were derived from an adult person with his or her consent. Also, as I mentioned, they can be derived from either a healthy person or from a patient who has, for which, I mean, who you want to cure. So, and it's, the process is minimally invasive. It's not like a very gross surgery kind of a process where you need to isolate those cells. It's simple skin biopsy through which you can obtain the skin cells or the fibroblasts, as we call them, and then introduce the factors, the stem cell factors from outside in a laboratory, and then culture them over a period of time to form the iPSCs, which can further be differentiated into any given type of cell, provided you provide them with the specific factors or specific stimulus. Now, how these advancements in stem cell biology actually happen? It never, what we have today, the advancements in stem cell biology, they were not there since the beginning. In the beginning, it all began like a simple 2D culture. 2D means two-dimensional culture, right? So that was a very simple culture with a single layer of cells growing on a surface, which we also call as a monolayer culture. Just a plain layer of cells growing on a normal culture ware. And these cells were, this 2D culture was actually, it's still being used in most of the laboratories in the initial phases. And it's a very good model to study various disease mechanisms. So if you want to find out how a particular, say, for toxicity testing, how a particular toxin can produce changes in the whole physiology. So you can pick up a toxin, expose the cells in the 2D culture to that particular toxin and see the effects it induces in them. So for a basic phenomenon like that, 2D culture of stem cells is still being used. But when we talk about an advanced disease modeling thing where we need more human specific results or responses, there comes into picture 3D culture or the organoids. Now 3D cultures were a more complex form of this cell culture of stem cells. That is like they are kind of self-organizing cell structures. So it's not just simple layer of cells, it's like many cells interacting with each other. They form networks with each other, they form connections with each other. That's exactly how the organs inside a human body are. Like they have, it's not just a simple single layer of cells, it's a network of cells. So 3D models, or the what we call as organoids, they came into picture because they better mimicked the human system or the human organs as compared to the normal 2D culture. So now we have like a lot of brain organoids, we have heart organoids, we have kidney organoids. So all these organoids or the 3D system of the stem cell culture is being used for basically for drug screening and disease modeling. So this was like 3D culture can be a bit tricky to maintain a lab in lab because it requires a lot of monitoring and a lot of maintenance. But at the end, you get a more relatable results. The more relatable to human system. But the researchers did not really stop there. They wanted a more complex system because 3D cultures were fine. They had the interacting cells, but they did not have the vasculature. They did not have the normal blood flow like we have in our system. They did not have like an exchange of gases. They did not have like solute transport that is normally happening in our body. So they took a step ahead and formed the organ on chip concept. What is organ on chip? Organ on chip is like the size. It's like the size of a microchip that houses a complete organ system. Now you have in this particular system, you have various channels coming in from different sides, which are lined with the living human cells, and they have all this gaseous exchange mechanism. You can have the solute transfer mechanism. You can monitor the pH of the system. You can monitor the peristaltic activity. If it's a lung system, you can monitor the peristaltic activity of the lungs. Then you can see the temperature, what the temperature is in the, through the sensors, you can assess the temperature of the system. So what you see here in this particular GIF is actually the model or the organ on chip model of a beating heart. That's actually a beating heart in the laboratory, not inside a person, not inside an animal. So this was pretty, whatever here when you have so many factors that are actually in the human body present in a model system, the results that the researchers obtained with this particular concept were like almost 90% identical to what you would see in a normal human being because it is a complete organ system, right? So this was a very, very important revolutionizing step in the field of disease modeling and drug screening and drug development. So now most of the bigger organizations that are into drug development, they are relying heavily on organ on chip. They're investing heavily on organ on chip concept so that whatever data they get that has human specific results, and there are no data ambiguities or the interspecies difference that are normally encountered when you actually do this process with a living animal system, because there are always interspecies difference between animals and humans. So that kind of the data that you get is not reliable enough in that case. So this concept of organ on chip kind of prevents that data ambiguity problem here. So that's one of the reasons why the drug development firms are now relying on this particular concept of organ on chip. So this is how the stem cell biology has advanced, and it's still advancing. Like people, researchers are actually thinking of going even like a step ahead again with combining all different kinds of organ on chip system and then kind of preparing a human on chip. I mean, this might sound a little crazy right now, but that's what the research right now is headed towards. So you never know. One day you might have a human on chip, so that you can assess the entire, human system, how it functions in a diseased condition, how it functions normally in like under normal physiological conditions, how the different organ systems interact with each other. So then maybe we can have even better picture of human responses towards disease and drugs in general. So why the stem cells are being favored in the pharmaceutical industry now? What's the benefit? The major reason behind employing stem cells in pharmaceutical industry or the drug development organizations was the accuracy of the results obtained and also the reduction in cost that is involved with the conventional drug testing methods. So here what is done is when you use stem cells, you have more human related responses as I mentioned earlier. So you don't have to worry about like the later stages of the trials. Like normally what is done in a conventional clinical trial of a drug or any toxin testing method, you have to first test them on animals, lower order animals, then it goes to higher order animals, and then it finally reaches the human subjects in the final clinical trials. So there is like 75 to 80% chances that whatever data you have in your preclinical trials with the animals, there's very high chances that you won't have the same effects in the human system because it's a completely different system from the animals. So when you have stem cells in the picture, you don't have to worry about that. You don't have to worry about the cost that whatever you invested in the whole preclinical trial phase stages, that might all go waste if it doesn't turn up like exactly how it's supposed to be in the human system, right? So with the advent of stem cells, this concern of the pharmaceutical industries was kind of reduced. They were relieved that they don't have to worry that much about the costs, and the results would be far more accurate as compared to the animal models, the conventional method that's being followed. Now, that's the reason why, for the toxicity testing, for identifying new therapies, these drug companies are actually relying on a stem cell model or a stem cell based system. Also, when you have human cells, you can be pretty sure of what the possible side effects could be of the drug that's being prepared, that's being tested. So you have a better picture of all the positives and all the negatives that a particular compound can induce in a human system. So that's another major important factor of using stem cells in pharmaceutical industry. And since stem cells, you can have an endless supply of stem cells. It's like they're pretty easy to derive from a normal person's tissue or a patient's tissue, right? So you can have an endless supply of stem cells with a more and that guarantee a more consistent method of testing, right? And then, as I mentioned earlier, this approach saves a lot of money since you don't have to worry about like your money all going to drain. I mean, if the preclinical data does not fit in the final clinical phases in the human subject. So you don't the pharmaceutical industries now they do not have to worry about that factor if they use stem cells as the model system. So how it's done. So one important thing that's being done in the pharmaceutical industry or the drug industry in general is deriving deriving iPSCs from a patient. So for example, here, you have a patient, say, of any particular disease that you're studying, or whoever like you want to treat. So you have a patient, you through a simple skin biopsy, you can isolate the fibroblast or the skin cells from that patient, then reprogram them into iPSCs through introduction of the stem cell factors. And now from here, when you have once you have derived the iPSCs, you can either directly differentiate them into a desired cell type. Here it's neurons because this is like the patient of Huntington's disease, which is a neurodegenerative disorder. So from here, you can either directly differentiate these iPSCs into a desired cell type and then form the final like intermediate. These are the intermediate neural stages. Then you convert them into final neurons, how they are, and then you can use these disease specific neurons for modeling of the disease, for drug screening, and for toxicity testing. So if you want to know how the disease functions, you can use these neurons because they'll have all the characteristics and all the hallmarks of that particular disease that the patient was carrying, right? So you can use them for disease modeling, for drug screening, and for toxicity testing as well. Another method of going about this is once you have the iPSCs, you can correct the mutations that are present in it, right? You through gene editing, that's one of the tools we have right now where you can introduce whatever the mutations are there in the gene, you can correct them. And then once these iPSCs have been corrected, then you differentiate them into a desired cell type. And these corrected cells or the neurons which are formed from these corrected cells can be transplanted back into the patient, which is what we call as the stem cell therapy right now. So there are two ways of using patient-derived iPSCs. Either you use them for disease modeling or you use them for stem cell therapy. Now, what their role in clinical trials is, once you have figured out how the iPSCs are used, where they can be employed, whether for drug screening, whether for disease modeling, that's all fine. But what interests the clinicians right now is how they contribute to the clinical trials, because that's the major concern these days once you're into the drug development industry. So earlier, the conventional method, what used to be followed was like you have a patient here, you identify the target against which you need a drug, right? And then you go for screening like multiple compounds. So you have to kind of like hundreds of compounds are tested against that particular target. And you get a lot of hits out of them. You might get a lot of hits. And then all those hits are then tested on animals. So like the number of compounds you have or the number of hits you have is directly proportional to the number of animals that you'll be using as the models for drug testing. And then after you get like some positive responses in the animals, then the final clinical trial stages, phase 1, 2, and 3 begins with the human subjects, right? So it was like a long process. A lot of time was involved, a lot of cost. The cost was high. And plus, the chances of success were less because you were relying on an animal system, which although shares like similarity with the human genomes and human genetic makeup, but is not exactly identical to it, right? So when the iPSCs came into picture, what happened was you still had the patient, right? You have the patient, you still had to identify the target, and you still had to screen the compounds. But what changed here was the compounds were tested on patient-specific iPSCs, right? The iPSCs which were derived from that patient and which were then converted into desired cell types. I mean, whatever disease, depending on the disease you're studying. So when the compounds were tested on these cells, the number of hits that were obtained were higher. Comparatively, you had more positive, like you had more positives, like instead of false positives. And these hits were kind of if they had similarity to any existing drug, if the compounds that were identified, they had some similarity to a drug that is already in the market, these compounds were directly used for the repurposing, like what you call as the repurposed drugs. And if not, if they had to go through animal models, the number of animals that were used for this particular process was reduced because you had exact positives that were to be tested. So you didn't have to use all the compounds on the animals for testing, right? You just use those hits which were like 100% positive, and you use them on animals, and then you used it on humans, right? So the number of animals reduced, the timing reduced, because it's always, if you have more number of animals, you need more monitoring. The more that, obviously there's more time taken. So here, as the number of animals reduced, the time reduced, that eventually led to reduced cost, and it also led to the concept of personalized medicine, which I'll be discussing later on. So you had more accurate results with this drug discovery method using iPSCs. So this is how the companies benefited when the iPSCs began to be used for clinical trials. Now comes the concept of regenerative medicine. Regenerative medicine is a part of stem cell therapy. It's a type of stem cell therapy, and it's like highly used now, and it's one of the major applications of stem cells. So by regenerative medicine, I mean that branch of medicine that uses stem cells to repair or replace a damaged tissue or organ or cells, right? So how stem cells contribute to that, how they can repair the damaged tissue in a patient. So how it's done is, suppose you have a patient here with some kind of spinal cord injury, say. So what you do is you take the skin cells, you have the skin fibroblast here, the basic skin cells we have. You reprogram them or convert them into iPSCs. You differentiate them into neural stems in a dish. Like this is all done in a lab outside the body. And then once you have your stem cells, the neural stem cells, you implant them or you transplant them back into the patient's spinal cord through an injection. And then it was seen that these cells started to grow or started to repair the damaged tissue. So it could be like through the replacement of the damaged tissue, or it could also be through the secretion of certain factors by stem cells that they secrete certain factors that can help in repairing the damaged tissue. So it was seen that some of the patients, like in the pilot studies, I'd say, the researchers or the clinicians could see the recovery, right? The damaged tissue. It was seen in one of the conditions of heart where there was a damage in the heart valve, I guess, and that was kind of reduced when the patient had the stem cells transplanted back. The patient's own cells were converted into stem cells, so there was no rejection because it was derived from his own cells. And the researchers or the clinicians saw a recovery or the repair of the damaged heart tissue there. So, but it still remains to be seen whether these stem cells replace the damaged tissue or they repair the damaged tissue through secretion of certain factors or growth factors that can help in the repair. So this is one of the most sought after application of stem cells right now, which is being used increasingly in the clinical aspect to cure various diseases and to avoid any kind of rejection or any kind of unnecessary immune response in the patients. Related with regenerative medicine, there was another concept that completely transformed the face of medicine, was the concept of personalized medicine. The personalized medicine is that when a patient gets the treatment based on his or her genetic makeup, right? It's like we are all humans, but we do not have exactly like identical genetic makeup. Like there are various people who have certain diseases, their genetic profile is going to be different from a normal individual. So when you derive the cells from a patient, you are essentially focusing on that patient's genetic profile, right? So whatever response would be would be emanating from that treatment would be specific for that patient, right? So there would be lesser side effects. There would be no rejection at all when such a kind of medicine is employed. So this is like, I mean, it's still in progress, the concept of personalized medicine, but a lot of work is being done on them. So the organ on chip concept that I explained earlier is one of the most important concept that's being used for uh, the personalized medicine concept here. So how it works is it works through microfluidics. As I explained, the channels are lined, the gaseous exchange occurs, those channels are lined with live human cells, and then you have a complete organ system here. So wherein you can test various compounds. So whatever results you get, they're going to be exactly identical to how that human being, or that patient is going to respond to that particular drug that's being tested, right? Because it is essentially from his or her tissue. So a lot of chips have already been derived. That there's a liver chip, there's a kidney chip, lung chip, brain chip. So, the Viz Institute in Harvard here in Massachusetts is highly involved in developing these organ on chip thing. And their next target is to, as I stated earlier, is to create a human on chip thing by combining various organ on chip or various organ on chips so that you have a connected network of various organ systems. So that would be like a major thing if it happens at all, because it's not as easy as it sounds, but it's not impossible either. So this is like one of the very important advancement in the stem cell biology or the stem cell research in the field of drug development right now. Now, since we've been talking about stem cell therapy, but if we have not had successful cases with stem cell therapy, then there's no point talking about them. So here there's a list of some of the diseases which have already been approved, for which the stem cell therapy has already been approved by FDA. Why I strongly emphasize on FDA approved stem cell therapy? Because whenever a therapy is designed using stem cells, it's very important that it gets an approval from FDA. If at all you come across any stem cell therapy that is not FDA approved, always avoid going to such a clinic. Like, this is a general information for anybody, because you never know what the quality could be, how effective or how safe it could be, right? So FDA approval for any kind of stem cell therapy is a must. It's a mandatory thing right now. So FDA approval was given to some of the FDA approval for stem cell therapy was given to some of the diseases, which were usually blood-related disorders till now, like leukemias, lymphomas, anemia, and various inherited immune system disorders. So these diseases, they already have been successfully treated with stem cell therapy. A lot of, in fact, there have been some FDA-approved drugs against, if I remember, against leukemia in that got approved in 2019 recently. Another is like this. Besides these diseases, there's so many other prevalent diseases in human population, like so many neurodegenerative disorders, metabolic disorders. So a lot of them are in clinical trials, and they're still being conducted to like they're being assessed for the side effects, for the efficacy, for the safety. So these diseases are usually the neurodegenerative disorders like Parkinson's disease or cardiovascular disorders or diabetes or stroke. So a lot of work is being done in these diseases too to see if stem cell therapy can work or can successfully work in case of such diseases. Now, besides just treating patients, the stem cell therapy has also contributed to revenue generation in a big way. So it's estimated that it'll grow with an annual growth rate of around 28% from 2019 to 2025, with almost an approximately reaching to a budget of around 167 million US dollars. And this is because most of the developed as well as the developing nations are now relying and are now kind of using stem cells for various research purposes in clinical trials, in therapy. So this is also, and there are a lot of stem cell banks being opened, a lot of trials going on for so many diseases. So all things cumulatively contribute to the global economy, right? So a huge portion, like 28% portion of there's an estimation like a 28% growth in the stem cell market. So they're not just treating the diseases, they're also contributing to revenue generation in a big way. Now, stem cells and medicine. So far, I talked about regenerative medicine. I talked about personalized medicine. I talked about stem cell therapy, basically. So it's all these three fields basically involve transplantation of stem cells or use of live stem cells as such. But there's another field, another concept that is the stem cell drug. Now, stem cell drug is not a very, I'd say, not a very new, not a very old concept. It's just like been introduced like 4 or 5 years back. And it's like off the shelf products of stem cells that can be used for treating a disease and that can have an industrial production, right? So, what are stem cell drugs? So, in if you talk about, since this is a pharmacy institute, so I know what everybody of you knows what a drug is in terms of pharmacology. It's a substance which can cure, prevent, or diagnose a disease, or in general promotes the well-being of a person, of an organism consuming that. And stem cell drugs are essentially off-the-shelf products which are based on stem cells, which can be used to treat certain diseases. So they are not essentially cells as such. They are some products or derived from those stem cells, which you can buy from a pharmacy, or like these are off-the-shelf products, right? And the cells that are from which these products or these drugs are usually derived are the hematopoietic stem cells or the mesenchymal stem cells. If you remember it from the earlier slide, hematopoietic stem cells or mesenchymal stem cells are the multipotent stem cells, or they're also known as the adult stem cells, right? So these cells, when you talk about stem cell drugs, it's usually these cells that are used in that particular concept. So what these products are, so it's like these stem cells secrete a lot of factors, a lot of growth factors, a lot of vesicles, vesicles or which we in biology call as exosomes. Now these vesicles carry a lot of biomolecules. They have RNA, they have proteins, they have microRNAs, they have hormones. So it's like it's a variety of biomolecules that are coming out of a stem cell, and they have multiple valuable properties. So what is done is these vesicles or these factors that are secreted by the stem cells, they are harvested, they are isolated, and they are